This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 107, recorded on July 2nd, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, which is in Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
But you're not in Charleston.
Now I'm in beautiful Waukegan, Illinois, home of Jack Benny.
How's the weather out there? Is it nice?
It's beautiful after the hot and humid Charleston. It's 63 degrees and relative humidity of about 60%. It's like springtime.
Nice.
They're actually complaining in Chicago that it's so cold for the 4th of July. And they're all going to have to wear coats.
Oh, well. Well, here in New York City, it's 27 Celsius, really cloudy. So my office overlooks the Hudson River, and, I have a great view. I'm very lucky. And today there are lots of boats on the river. People are already out there taking the day off, I suppose.
Or staking out the territory for the fireworks.
Yeah, that's right. But here we are working. We're doing a podcast for you, and it's just Michael and myself today. Our colleagues are off. They decided not to join us for various reasons.
I think we should have did this podcast yesterday, and then we could have had the palindrome of, since this is episode 107, we should have recorded it on 701.
Boy, that would have been something. Is that the sort of thing you notice?
I just noticed it looking at when you said 107, and I said, today's the second.
It is. Coming up on, yesterday, as you know, was Canada Day, right?
Correct.
They celebrate the unification of Canada. And July 4th, of course, is Independence Day in the U.S. We celebrate, I guess it's the signing of the Declaration of Independence. Is that correct?
Yeah.
On July 4th, 1776. And everyone goes crazy here in the U.S.
And I remember the bicentennial in 1976, and everybody went crazy even more so.
It's funny. I was going to point that out. I actually went to Washington for the bicentennial.
Oh, you did?
Yeah, and I sat on the Great Lawn and watched, I don't know, a concert, fireworks, whatever. It's interesting. I was with a bunch of people. I remember this very distinctly, and this has to do with one of our papers today. There were a bunch of guys and girls, and I was very young in 1976, and some of the people, one woman said, I need to go to the bathroom, and I said, well, I think the porta thingies are over there, porta potty thingies, right? So she went, she came back and said, the line is a mile long. We had this blanket, right?
Uh-oh.
And, in about 10 minutes, she was sitting on the grass. She moved from the grass to the blanket. And I looked at her, I said, did you? She said, yeah. She just went in her pants because she couldn't wait. And she didn't want to wait online. And today we have a story in urine. That's why this is a relevant story. I always thought that was cool that she would do. By the time she walked home, it was dry, so it didn't matter. Yeah. It worked. We have a snippet in a paper for you today and some email, and this is going to be a 12-hour episode because Michael, always likes to talk, and I don't restrain him in any way. So we're going to go on, and maybe by the time we're done, there'll be fireworks, Michael.
We can only hope. We can only hope.
A snippet. I have a snippet, which is a paper that was just published in Science Advances. Is that the name of the Sci Advances? Yeah. And this is called, it was just published in April of this year, The Microbiome of Uncontacted Amerindians. First author is Jose Clemente. The last author is Gloria Dominguez Bello, and some of the names you may recognize on this paper, Rob Knight.
Of course.
A man who wants to sequence the world. I presume he lent his bioinformatic expertise to this. And this paper, Michael, is a PDF, and it chokes all the computers I've tried to scroll it on because the figures are so dense and so complicated. I can't scroll through without it getting. Did you happen to notice that or not?
I did. I did, and I have a pretty good internet connection at the office, and I'm going, oh no.
It's really strange. Anyway, this is all about the microbiome, which we talk about all the time on TWiM, and we know that it's got important roles in human health of various sorts that we have talked about in many ways on many episodes. But the thing is, we only study the microbiome of westernized people.
Mm-hmm.
People who use a lot of antibiotics, hand sanitizer, and do all sorts of things to kill microbes, right? So the real question is, what would a microbiome look like in a person who never saw an antibiotic? And how has it changed? And that's the question that's addressed in this paper. And, I never thought of it. It's brilliant, right? I mean, of course, there's some people left on the globe that don't have westernized habits. And the one they used in this paper is the Yanomami. All right. These are individuals living in the Amazon forest. In this particular paper, they're from Venezuela. These are people who live semi-nomadic hunter-gatherer lifestyles. They have no contact with anyone who's not a Yanomami. They don't use antibiotics. They don't use hand sanitizers.
Soap.
They don't use soap. They use toothpaste. They don't have to wash their hands after preparing food and going to the bathroom because they just go in the woods probably.
Mm-hmm.
And just so that you know, this particular group of individuals that they sampled in this paper, it's from the high Orinoco area of the Amazonas state in Venezuela. This is about an 80,000 square kilometer area of jungle, which is a homeland for 15,000 Yanomami. Most of them are living near rivers, but some of them are away from rivers in mountainous areas. And in 2008, an unmapped village was sighted by an Army helicopter. And the next year, a medical mission landed there. One of the authors of this paper was at the site, and they collected samples from these villagers. And we don't know where this is, and we don't know their names. But these individuals were all hunter-gatherers. They have no agriculture. They don't domesticate livestock. And, of course, they don't use antibiotics. They got a number of samples: mouth samples, 28, forearm skin, 28, 12 fecal samples. And this is from 34 of the 54 villagers. So 54 people live in this little group, and their ages were between 4 and 50 years old. So they did microbiome sequencing by sequencing 16S ribosomal RNA, and that's the topic of this paper. What did they find? Really cool things. First of all, their microbiome has the highest diversity ever reported in any human group. And that's for the feces and the skin. And this compared to the U.S. microbiome. Interesting, the mouth was not as diverse. It was more similar to ours.
Well, that makes sense because it is, as it were, as I tell my dental students each January when they start, it's a cesspool. I mean, we're constantly feeding it, and we would expect diversity, and we're only really washing it at best twice a day with toothpaste.
But the diversity is like that of the U.S. So you're saying that we have a very diverse oral microbiome because it's a cesspool. Okay, got it.
Yeah, it's a there. The concentration is about 100 million.
Yeah, but I would think that we're putting different things into our mouths so that that would influence it. But apparently not. And also we use toothbrush and toothpaste and mouthwash. They just rinse their mouth out. So,
yeah, but most of the microbes in our mouth are physically attached to surfaces,
so that any washing is not going to change it. Huh?
It's really the abrasion that does it. And if they're like most indigenous peoples, they probably are using something to preserve their dentition, because that's an old behavior. Because if you couldn't eat, you failed to reproduce, and so taking care of your teeth is one of our older behaviors.
Right. Next, they have the lowest variability from one individual to another. So, for example, in the fecal microbiota, they share a higher proportion of the different types of bacteria compared with the U.S. They go into differences in the various taxa, and I don't want to go through that, but I wanted just to point out one. in the U.S., when you look at fecal microbiota, you don't find Helicobacter, but you do find it in the Yanomami. Now, what does that mean, Michael? We've gotten rid of it with antibiotic treatment?
Well, no. Remember when we did the early TWiMs on Helicobacter, we learned that the technologically advanced societies of the globe started to lose Helicobacter, likely with the advent of indoor plumbing back in the turn of the 19th to the 20th century. And they were tracking Helicobacter because it's one of those vertically transmitted microbes from caregiver to infants. And they learned, I think, if I'm remembering right, it was either one of the Scandinavian countries, I think Norway, that first described it. And they found it began at the turn of the 19th to the 20th century. So they were likely attributing it to the advent of indoor plumbing.
Interesting. Which obviously the Yanomami don't have, so they have Helicobacter. I wonder if they have less asthma than we do.
They probably do.
Be an interesting study to do. Of course, you'd have to hang out and observe them for a while, I suppose. All right, next, they decided to isolate bacteria from these samples to have an archive of strains that could be used to study. They took these with them. They isolated 1,097 bacterial strains from fecal or oral samples, and they characterized 24 E. coli strains using multi-locus sequence typing based on 7 housekeeping genes, and they showed that these strains of E. coli differ substantially from those that are circulating in Western societies, and none of them belong to a phylogenetic group called B2 that's common in Western populations. So it's not surprising because these are isolated communities. They're not sharing the same communities of bacteria. The interesting part, though, is that all of the E. coli isolates that they made from these individuals were sensitive to 23 different antibiotics that were tested. All 23 antibiotics inhibited the growth of all of these E. coli isolates. There's 24 different E. coli isolates. So next, they did some culture-independent analysis saying, are there antibiotic resistance genes? So they took the DNA of these isolates. They took 38 isolates, and they would take the DNA, put them in vectors, and then ask, are there antibiotic resistance genes in the genome? Because we've already done culturing, and we can say that they're sensitive. So in this way, they can find antibiotic resistance genes in the genome of these E. coli isolates. They found 28 different functional resistance genes, including resistance to synthetic and semi-synthetic antibiotics, which they can't have ever been exposed to. Because those are right? They've been made by humans elsewhere in the Western world.
The bacterial world is getting prepared because these peoples are 11,000 years removed from us.
Yeah.
So antibiotic resistance genes were preloaded into the genome of the microbe 11,000 years before they needed it. Talk about pre-planning.
Of course, Michael is being funny.
No, but it really is.
We don't want people to think that there's any kind of planning.
Oh, no. No, we'll get letters and I'll get in trouble.
Yeah, no, they are probably there because in nature, bacteria encounter each other and they make antibiotics and they're ready for that. Now, the interesting thing here is that these genes are not expressed because otherwise the E. coli isolates, the bacteria in the culture-dependent assay would have shown antibiotic resistance, right? So they say they're actually near mobile elements like transposons and plasmids, transposons and phages. So if you treated these individuals with antibiotics, you could then probably the DNA would rearrange to make these resistance genes expressed, and then you'd have resistance. So as Michael said, they're primed and ready to go.
And it's the rainforest. This is a rainforest peoples, and the rainforest, of course, is a great source of diversity in terms of microbes, and a lot of the microbes of the rainforest do indeed produce antimicrobial material compounds, and with the antibiotic resistance gene, you can almost view it as an antidote against the germ warfare of the jungle.
That's right.
The microbes are making these compounds for their own use.
Exactly.
And these antidotes are likely coming in with their diet because, they are people that cook, but they're probably eating a lot of foods au natural, and they're not scrubbing them with soaps or lower pH water or things that would scrub the bacteria off.
Right.
And so the whole genetic exchange that goes on in our guts is probably occurring in theirs as well. And the genes come in, the genes go out based on the selective need of the community that's there.
Yep. Now, when you look at the sequences of these antibiotic resistance genes, you see that they are homologous to those you find in Western industrialized societies. So they call this the resistome, this collection of antibiotic resistance genes. It's similar in the Yanomami and the Western countries, despite different antibiotic exposure histories. Next, the gene. So they speculate, where did these genes come from? And they speculate, as we've just sort of touched on, they may have come by exchange with soil microbes. They're close to the soil. They probably pick them up all the time on their skin or in their food. Or they may have traded with other populations that have received antibiotics. So, there's probably a chain of trade. It starts way upstream and then somehow gets into a Yanomami and then makes its way to this isolated population. So they could have arrived that way. But either way, these people are not getting antibiotics, so they're maintained there without antibiotic selection. 14%, as I think I mentioned this, 14% of the antibiotic resistance genes are near mobilization elements. So, again, if you treat with antibiotics, they will likely move around and become active. Assuming a doubling rate of once per hour of bacteria, an estimated 100 million bacterial generations would have likely occurred with little gene migration in the microbiome, moving around of genes. And this is compared to much higher rates in the westernized world. So having these strains will be really nice to study the evolution, right? So you can take these strains back in the lab, you can put antibiotics, you could look at how DNAs move around to turn on these antibiotic resistance genes. So it's a really nice collection that they've made here. And
it really gives you a sense of how valuable something as straightforward as 16S ribosomal RNA sequencing adds to telling a story of an isolated group of humans and how they have survived, and the ubiquity of the microbial world and how it adapts to us and we adapt to it. And you can learn many different things.
Mm-hmm. All right. Well, one more thing. These samples were picked up when this village was first discovered. So a medical team went in, got these samples, and they introduced antibiotics.
Of course they did. And and I think they also, if I'm remembering reading the paper right, they vaccinated the children as well.
Yeah. So I guess you have an ethical obligation to do that. I'm sure they went in and there were some medical issues for which they treated them with antibiotics. So I just thought it would be interesting to go back and see what happens, right, with the microbiome. Is it really going to change? Is the resistome going to change? Are these genes going to be activated? I mean, you don't want to do experiments on people, but, I don't know if this antibiotic is a one-time thing or they're going to have continuous bringing antibiotics there, but
well, it's really the good way to do the experiment, because unlike you and I, where we really don't know if we're getting antibiotic-free food, for the most part, their food is indeed free of any man-made antibiotics.
Yeah.
And I think it's our food supply that is really sneaking antibiotics into us, even if you are making a conscious decision not to demand an antibiotic prescription when you feel unwell and it's likely a viral infection and you really don't need the antibiotic to clear the infection. And antibiotics are good when you do indeed have a fulminant infection for which you need to be treated. Like last the last TWiM episode when we were talking about Francisella.
Yeah.
But here, I think it's really the ideal way, because if they were indeed given that one course of antibiotics, Rob Knight could go back in and say, okay, it's been X number of years. Is their population in their at least fecal samples stabilized? Is it back to where it was, or did we lose anybody important?
Yeah. And I'll end up with the last line of this paper, which I think is great. They say, quote, causation and functional consequences of microbiome changes need to be understood before functional restoration of the microbiome is possible to attempt to reverse the current global trends in metabolic and inflammatory diseases. What do you think of that, Michael?
Profound.
I mean, everyone wants to get fecal transplants and modify your microbiome, but do we really know the consequences?
No.
We don't.
And it's really the law of unintended consequences has really played out in this antibiotic era that you and I have grown up in.
Mm-hmm.
Because you and I were children of the 50s and 60s when antibiotics were in their heyday, and they were really the miracle drug. And the 90s and 2000s, antibiotics are now still miracles, but you have to be very judicious in your selection of the antibiotic. In the old days, you could just write a prescription for penicillin and it would remarkably work.
Yep.
And today, unfortunately, that's not the case. So We'll see what happens. As Elio sometimes says, stay tuned.
Is that what he says? I thought you said. I think you say that sometimes too, don't you?
I do.
So this is Science Reports. Is that the name of the journal? Science Advances.
It's put out by
the AAAS, right?
AAAS.
What is this? Is this? I haven't encountered science. I know Science Reports are the open access things, right?
Yeah.
What is Science Advances?
It was new to me the first time. So it's part of their journal. They have this family of journals. They have Science Translational Medicine, Science Signaling, and this is their new of their flagship with the word science in the journal. This is Science Advances.
All right, here it is. Their first open access online only journal.
Oh, so this is like MBio of ASM.
That's right. Open
They
access.
Copied us.
And online only. Yep. Broad array of fields, significant research, all the usual stuff. Well, what am I thinking that there was another? I guess this is the open access one. I think I'm talking about the short one, which is Brevia, maybe, which is not a journal. It's just articles. Okay. Yeah, this is maybe the first one I've seen from this, journal. But MBio has done very well, as you know, so there is clearly a need. Open access is great. I think it's important that all people get to read an article if they want, not just scientists. And I know that there's always an issue in paying for it. So ASM has lots of for-pay journals still that help support MBio, right? But then, of course, the page charges are substantial, and that helps as well.
Yeah, one of the other authors of this paper was the third author on the list is, of course, the ASM lecturer from this year's general meeting, Marty Blazer.
That's right. I was looking. I knew I saw his name, and I couldn't find it this time. Marty Blazer's on this paper as well. Yep.
Yeah. And it was principally authored by Jose Clemente, was the first author, and then Erica Fearson and Marty Blazer, Koldip, Sandhu, and there's a whole long list of others.
Yeah, I should probably point out that as soon as I scroll to the top of this. So, of course, Marty is at NYU Medical School here in New York. They got some people from Mount Sinai here, Washington University, some institutes in Venezuela, Caracas, University of Nebraska, Puerto Rico, University of Colorado. So it's quite the collaboration, as we have seen in a lot of these papers. All right, that's our snippet, which we went a little over on, but that's okay because we don't have the policers here.
Yes, the policers. And I'll try to be brief with our next paper. Our next paper has received a lot of popular press. It appears in the Journal of Biological Chemistry, and I'm going to try to do justice to the biochem, but not go overboard. So It's a really interesting paper. The title is Human Urinary Composition Controls Antibacterial Activity of Siderocalin. And it was authored by Robin R. Shields Cutler, Jan Crowley, Chia Wang, Ann Stapleton, Courtney Aldrich, Jonas Marskal, and Jeffrey Henderson. And they're from the Division of Infectious Disease, Center for Women's Infectious Disease Research, and the Department of Internal Medicine at Washington University in St. Louis, Missouri, as well as the University of Washington in Seattle, and the Department of Medicinal Chemistry at the University of Minnesota, Minneapolis, and the Department of Infectious Diseases at Bern University Hospital and of the University of Bern in Bern, Switzerland. So again, we have an interdisciplinary team of infectious disease physicians, microbiologists, as well as biochemists contributing to this paper. And so here's the story in a nutshell. During urinary tract infections, humans secrete this protein, siderocalin. And as you have learned in past TWiMs, anytime you hear the word sidero, it typically means it's binding a metal. In this particular case, it's iron. And this is to block the bacterium's ability to harvest iron from the location that they happen to be in. In this case, it's the bladder. And the next point that you have to keep in the back of your mind as we go through this is human urinary pH and metabolite composition of that urine strongly affect the siderophore or siderocalin's antibacterial activity. And the conclusion that they have reached is siderocalin uses a subset of urinary metabolites as cofactors to withhold iron from E. coli in competition with the universal bacterial siderophore produced by E. coli called enterobactin. So the reason I picked this paper is because urinary tract infections are a big deal to man or to humans. Urinary tract infections are the most common bacterial infection in women, with over one half, and this is a really important distinction here, one half of all women will experience at least one urinary tract infection in their lifetime. So, 50% of the planet, 50% of the humans are effectively made up by women, and one half of those folks will actually get a UTI. So a quarter of the globe's population will get a urinary tract infection in their lifetime.
Remember the fellow from Minnesota we had on a TWiM at ICAC or AGM?
Yes, I do indeed.
That's because he gave a great talk about this, and we had him on. I think we had him with Jim Gurn, right? Yes.
Correct.
So let me see if I think it was, yes, URI and UTI at ICAC. Mm-hmm. That is TWiM number 64. James Johnson.
James Johnson.
We had James Gurn and James Johnson.
We had the two Jims. One was Jim and one was James, if I'm remembering right.
So we had UTIs, which is the subject for this paper, and URIs.
Yes, upper respiratory infections.
Yes, that just rang a bell.
So
sorry about that, Michael.
That's okay. So this infection of the urinary system, or uncomplicated cystitis, which is an infection of the bladder, is principally caused by E. coli. The thing, 86% of the UTIs are E. coli, and 4% of them are Staph saprophyticus, and then there's a slew of other gram-negatives that make up the remainder. So it's principally E. coli.
Which comes from the gut tract, right?
Comes from our own digestive tract. And that was what James Johnson was telling us about, is that, some of these deadly urinary tract infections are actually re-inoculating the women's vaginal flora, and then it contaminates, if you will, the plumbing, and the net consequence is they succumb to this urinary tract infection.
Michael, the design is really bad. Okay, you have yes. Where fecal material exits, and then you have the urinary tract very close by, I think James said that in samples of healthy women, they always find E. coli contaminating the whole region.
Right.
And of course, men don't have as much of an issue because the urethra is longer, right?
Correct.
And so that prevents. But as men age, then you start to get it because your immune systems get compromised.
And your flow, because of the prostate enlargement, your flow does not discharge as much, and you get a biofilm, and it becomes a significant event. And to put this into dollars and cents, there are about 7 million office visits per year in the United States that are associated with about $1.6 billion in cost. So this is not cheap in terms of understanding it. So I think the reason I picked this paper is because I like iron. As a graduate student, I grew up in a lab that tried to understand iron, and it really intrigued me of how these authors took this problem apart. So it's really a fascinating story, and we had the Kipling quote on in the past, but I'll just tell it again because the Kipling quote or Kipling poem tells the story of a rebellion by a baron who fails but is ultimately forgiven by a king. And that's effectively what we're going to discuss here with E. coli and this whole world of iron. And the way the Kipling quote goes is, "Gold is for the mistress, silver for the maid, copper for the craftsman cunning at his trade. Good," said the baron sitting in his hall, "but iron, cold iron, is master of them all." And so as we begin to take this poem that was published in Rewards and Fairies in 1910 as an introduction to the story of the same name, we consider this whole story of the importance of this micro trace element, iron, and how it can really control the ability of a pathogen to get a foothold. And in their introduction, the authors postulate that it's this differential susceptibility to sequestration by the innate immune defense protein siderocalin that is really driving a lot of what goes on in the bacteria, and it has been principally proposed as a major selective pressure driving acquisition of these chemically diverse virulence-associated urinary pathogenic E. coli siderophores. And in one of their first illustrations, the authors go into, because this is a JBC paper, so they have to teach us some chemistry. And so they give us the charismatic acid pathway, which is the precursor molecule to these siderophores, and they teach us how to make a siderophore. Those of you who forgot your charismatic acid biochemistry, and here's where Michelle's eyebrows go up, and I get the lecture.
You don't have to worry.
But I'm just going to say it's erythrose 4-phosphate plus phosphoenolpyruvate, I believe. I don't have cheat sheets in front of me. I'm just remembering the biochemistry I was taught as a graduate student as Indiana. The charismatic acid pathway, you have the Ent-B gene complex that will convert charismatic acid into enterobactin, which is the constitutively expressed virulence factor by E. coli that binds iron and enables it to grow in our bladder, which is why it's responsible for 86% of the urinary tract infections. But E. coli is clever. In addition to Enterobacter, it has acquired via horizontal gene transfer 2 other siderophores. One is salmonellin, and you can guess where it came from. It came from salmonella. And the other is Yersinia bactin. And the pathway that gave us enterobactin, there's one gene that is between enterobactin and the salmonella siderophore, and that's called the IRO-A gene, and that gene product transforms it into salmonellin. And then in an independent pathway from charismatic acid is 2 gene products that make the Yersinia bactin. So good old E. coli constitutively makes enterobactin, but then it can turn on and make these other two siderophores.
And it would do that if iron is limiting? Is that the idea?
And they would do that when iron is limiting.
So, Michael, in the gut, which is the normal place for E. coli, right?
Mm-hmm.
I presume it also gets iron via enterobactin, right?
Correct.
So,
And
it's actually in the urine, and it's in the bladder accidentally. It doesn't. It's
correct.
It's not as niche. So, if iron becomes very limiting in the bladder, then it needs to. So, it's got this extra backup, which must come into use in the gut as well, right?
Right, because it's the transit time, and it's how you eat food and it comes out the other end, and there's a specific transit time. Since this is the 4th of July weekend and many of us eat corn on the cob, you can do this experiment at home. Eat the corn on the cob and then track your stool.
If you eat it with spice, it'll come out faster, right?
And sometimes, yes.
That's what I've been told, that spicy, like sauces
Yes.
and so forth, increase the transit time, right?
So citrocalin is introduced to the urinary tract by neutrophil granules and our uroepithelial cells, which regulate its concentration by 100-fold within 24 hours of the bladder sensing or perceiving an infection. And so 100-fold, you don't have to do any fancy statistics. You just can measure it. And
it's my kind of result.
Yeah, it's my kind of result too. And so citrocalin restricts iron's accessibility by binding the iron 3 complexes with high affinity with the protein's positive charge binding site, and they called this the calyx. And this positively charged binding site binds this iron 3 complex, and it actually steals this iron 3 complex that's already bound to enterobactin. It can steal the iron-laden enterobactin, and citrocalin will bind it. And so it can actually steal it from E. coli if the pH and the other things in the bladder mixture are right. And so that's what we're going to do as I take you through the data. The paper is very straightforward, but there's a tremendous amount of data. They gave us 11 figures. This is a full-blown JBC paper. And what they did is they used genetic and chemical approaches to ask the following. How do urinary pathogenic E. coli siderophores and citrocalin interact in a chemically distinct urinary environment during this human infection? And they learned that human urinary tract constituents make enterobactin critically important for resisting citrocalin. And so it's sort of the counterintuitive. You would think that citrocalin would be in charge of everything, but in reality, the enterobactin has co-evolved with our ability to fight off these infections, and it's really a function of the jumping towards the end of their paper. And when they take us through their summary, they argue that it's pH and aryl alcohol constituents that actually are the business end of all of this. So first up, they demonstrate to us that citrocalin is significantly elevated in women with uncomplicated cystitis. So it's a straightforward experiment. They collect urine from women that have an uncomplicated UTI, and they have a control group, and they ask a simple question with an ELISA, how much citrocalin is present. And what they learned is that the levels of citrocalin were higher in women with cystitis than in women without. And but what they noticed, and they used something called 2K box plot, in which the average is displayed in the horizontal line, and a 2K plot is the median, and the mean is indicated by the plus. And so what you see is the variability that is seen, but it's still significant. Women with cystitis have higher citrocalin. So the human bladder surface is coated with this, and this anionic glycosaminoglycan layer of our bladder surface actively attracts siderocalin, which is a cationic protein of an isoelectric point of 9. I told you it was a biochemistry paper, so we'll give a little of their great biochem data. But what really sets their story apart is they had a strain of E. coli that causes bladder infections, UTI, urinary tract infection 189. And all of their experiments start off with a common thing. They start off with an inoculum size of 1,000 colony-forming units per mil. And they also had mutants that had siderodeficient mutants. They either were delta enterobactin, and it was the ENT-B gene, which will knock out both enterobactin and the Salmonella siderophore, or they had a mutant with the IV that will knock out the Salmonella one, or they had the one that would knock out the Yersinia bactin. And so they had these mutants ready to go, and that's the way they took us through the story is using the mutants. And they did the bulk of their experiments in a chemically defined medium that was also scrubbed for iron, because a little bit of iron can go a long way. And they learned that the siderocalin limited the growth of the wild type UTI 189 in its ability to limit growth was greatest in the mutant strain that only expressed enterobactin as its sole siderophore to steal the iron from siderocalin, and the smallest effect was in the mutant that was expressing just Yersinia bactin as its sole siderophore. So I always love experiments with mutants because they're so clean. It's a black and white event, and as you look at their particular figures, you just see how remarkable it is. Even though they did statistics on it, you can just look at their plots, looking at the log colony forming units per mil on the y-axis, and then looking at plus and minus the 1.5 micromolar siderocalin versus PPS, and you can say it's night and day. It's so easy to figure out, and as they look at the various mutants, you can actually see what's actually going on. And so these results then show that urinary growth conditions substantially can alter previously described relationships between siderocalin and E. coli siderophore biosynthesis, because they had the 3 different mutant families. And as they go through it, they simply ask the question by measuring the log fold inhibition by siderocalin being present. And as they take you through the figure, they present the wild type data, and then they have another experiment in which they have pooled urine from individuals. And
so we got this defined medium that you mentioned, right?
The defined medium.
Scrub divine, and then they also grow the bacteria in urine, right?
In urine.
Okay.
And so that's their comparison.
The urine is from UTIs?
Yes, they get them from, they get unpooled urine specimen from donors that are, and this is where it gets a little confusing because they have so many different urines. They have some urines from donors that do not have UTIs. So let me check the figure legend to make sure I got that right.
I see. They originally get them unpooled, and then they make pools for some of the experiments.
Correct.
So in this one figure, they got pooled, which shows you how the taking away the siderophores, and B in particular, is really bad for growth. You get a lot of inhibition by urine.
And the inhibition is measured as 3 orders of magnitude, 10 to the 3-fold inhibition.
Yeah.
And whereas if you look at some of these others, it has no effect whatsoever. So NB was the master anaerobac, and that's what led them to consider how important Enterobacter or Enterobactin was to the process.
So Michael, if you look at these individual urines,
which is immediately below in this particular figure.
And they can assay the growth of bacteria in individual urines, right? Not pooled.
Right. And they had 50 donors.
Right.
They had 50 donors, and they talk a bit about specific gravity. If you're not familiar with specific gravity, it's a really simple and straightforward test in which they just measure your urine gravimetric weight against that of water. And water is operationally defined as it's effectively measuring how well your kidneys are processing it. So the more water you drink, obviously, the more likely your urine will be similar to water. And it really goes through it, and you can measure that. There's a normal level of specific gravity of urine between 1 and then 2 zeros and a 2 to 1030, and that's at normal. When your specific gravity goes up to 1.01, it suggests you're mildly dehydrated. And then if you begin to spill glucose into your system or protein, it will obviously raise the weight, which will suggest to your healthcare provider that your kidneys aren't up to scratch and doing what they need to be necessarily doing. So they're very much into looking at the importance of it, and that's what gave them a lot of clues as they began to work out the story associated with urine, which and the quote is, urine is remarkably complex and a variable biofluid. And they suggest that metabolism in the intestinal microbiome is responsible for shaping the chemical composition of our urine because of its multiple idiosyncratic sources: what you eat, how much water you drink, how well your kidneys are filtering, and of course your comorbidities, whether or not you're a type 2 diabetic, a type 1 diabetic. All of those things can fall into the system. And so that figure that has lots of little dots on it is measuring the log to the base 10-fold change in colony form units from the inoculum. And so they're starting with 10 to the 3 bacteria, which is log 3. And if they're measuring how many logs it will go up, if it goes up to log or 6 on their scale, that means that's a concentration of 10 to the 9 CFUs that have increased, because that's effectively what they're measuring. And again, what they learned is disrupting biosynthesis of enterobactin, but not salmonellachalin or Yersinia-bactin, greatly increases the siderochalin's urinary antibacterial activity.
What I find cool about this plot is the one where they look at individual urines, right? And they classify them as either restrictive or permissive for E. coli growth. They actually use, this is a ratio of the growth of wild type over the Enterobactin B mutant and how they respond to siderocalin. So, if you are restrictive, then siderocalin response is not going to be as great. Whereas if you're permissive and you add siderocalin, it's going to inhibit it quite a bit. So that's what I like about that.
And it's a real easy way, and that's one of the reasons I picked the paper, is it was a really straightforward visual for me to understand the importance of the urine and how it is contributing. And I think that's why it got a lot of press too, was this one figure or this one reduction, one way that they reduced their data showing permissive and restrictive. And as the snippet you talked about, Vincent, where you were talking about diet and presence or absence of antibiotics. This is a really interesting thing as we begin to tease out the remaining data of the paper, where they begin to ask questions about simple things of urine like pH, and they ask about other metabolites in urine. And you can then simply look at these ratios of the wild type growth plus the serocalin, or the delta ent B, which knocks out the enterobactin and the siderocalin, or not siderocalin, the salmonellacalin, and plus and minus the siderocalin added, and you can figure out this restriction. So as you begin to tease apart the fractions that are in, as they begin to fractionate the urine and ask the question, what's in it, they were then able to figure out what was important in urine, to understand the importance of these parameters in urine. And if you're trying to come up with a non-antibiotic way of treating a urinary tract infection, it may suggest a strategy to modify diet, to modify behavior, to effectively help you get over your urinary tract infection, or simply to prevent it.
What I found really interesting about this plot of individual urines, 50 different ones, whether they're restrictive or permissive for growth of E. coli. So the permissives are all pretty much in a very tight range in this activity measure, but the restrictives are all over the place.
Yeah.
They range from the slightly to the intermediate to the highly restrictive, right?
Yeah, and that these are from 50 individuals, and they got them from men and women that did not have a UTI.
So clearly, as you said, urine is complicated, and whatever it is that is restricting, there's a wide range in the human population.
Mm-hmm.
It's interesting.
So we've only gone through the first 3 or so figures, and and there's 7 more, and I don't want to trivialize the data that are in the other 7 figures, but it pretty much is asking the question, what's in the urine, and does pH matter? And so I think what I'm going to do is jump to, and I hate to use the word figure 6, but that's where I'm going to go to if you're following along in the paper, where the title of the figure is Urinary pH Distinguishes Restrictive from Permissive Urine. And so what they learned here is that the pH was very significant, or not, that can't be very, it is significant or it's not. Otherwise, I'll get notes from the statistician. It was significant. And when you had a pH of 6.8, it became restrictive. And when you dropped it down to 6.0, it was then became permissive.
So they measured the pHs of these individual urines, right?
Correct.
Okay.
And they did a plot looking at the siderocalin activity at various pHs, and they neutralized the urines with either sodium bicarbonate to raise the pH, or HCl to lower the pH of the urines. And they measured the urine siderocalin activity, the specific gravity of the urine, and there wasn't anything remarkable about the age, the donor who was supplying the urine, the serocalin activity in nanomolar concentrations. The specific gravity wasn't significant. The iron concentration wasn't significant in the urine from the donors, nor was the copper concentration different in the urine. It was just simply the pH.
So higher pH is
better for restrictive urines.
Got it. And lower pH allows the bacteria to
get the iron. And so,
and these are siderocalin dependent effects, right?
Correct.
All right.
And so then they began to tease apart what was the magic stuff in the urine. And they did this via the newfangled mass spec.
So, Michael, why do they think that it's not just pH?
Because the pH was varied, and they talk a little bit about that in the discussion.
Mm-hmm.
And do you want to bring up your discussion with the first author? Did she tumble to that?
He.
Oh,
no,
he.
No, but I think so. The way the paper goes, they just said to look for something beyond pH.
Correct.
We did the mass spec that you're going to talk about. I just wondered if you had any insight into why. So if you find that pH, so you observe a correlation of high pH with restriction, then you manipulate the pH with bicarbonate and you confirm that. I don't know. I don't know why you'd look for something else unless you want to know what the pH is doing, I suppose. Right. So you assume that the pH environment is making something else active or not. And so that may be it. Right.
Yeah. And I think it's because they did that rock curve in which they looked at the rock curve using the 50 specimen data set. And I think it was the rock curve that probably forced them to think that there was something else in the urine.
Okay.
And what they did is they began to analyze the urine via MSMS, which is tandem mass analysis, and they found a prominent 80 atomic mass unit neutral loss from all three metabolites, corresponding to a sulfite neutral loss observed from the MSMS of the sulfonated aromatic alcohols or enols by a group, first author Yi, that had published on it. And they did further analysis by HPLC and then LCMS, and they then began to discern what was magic in that urine, and the urinary metabolomes distinguish restrictive from permissive urines. Again, because they had that cool little test of being able to have this mutant and the wild type, and they had this ratio, they were able to discriminate from this. And so they had a urine fraction, they had an aerial sulfatase fraction, they had the urine fraction plus the aerial sulfatase fraction, the paracreosol fraction, and the metacreosol fraction. And as they began to distinguish it, they learned that the citoracalin antibacterial is associated with urinary aerial sulfates.
So so I think a lot of people probably lost you, but the bottom line is when they're looking for a compound associated with restrictive growth, it's an aryl alcohol, right?
Correct.
What is an aryl alcohol, Michael?
It's what gives that urine smell as you walk the streets of Charleston from our horse-drawn carriages.
Really? Is that it?
I believe it is.
Huh.
It gives the distinctive horse-drawn carriage urine smell. It's that sulfonated aryl alcohol because it's quite noticeable to the nose.
Oh, and it's a rather simple compound, isn't it?
Uh-huh.
Let's see. I'm going to look it up here. It is a no, I got the oxidase. I don't want that. I want aryl alcohol. I want to know the structure. Why am I getting
What's
the
Google
oxidase?
doing for?
Okay, here we go. Aryl alcohol is it looks like a ring with 3 double bonds. So that would be a benzyl ring, right?
Correct.
With a couple of side groups. Yeah.
Mm-hmm.
It's
very
simple.
aromatic.
Yeah.
Very aromatic.
It's very aromatic. Michael, you're either aromatic or not, right?
Well, no. You smell it, that aromatic, not aromaticity that you get from the pi orbitals sharing each other from chemistry.
Right. Aryl alcohol. So that correlates with restrictive activity, but at high pH, right?
Yes. Well, not really high pH because 6.8 isn't that high.
Yeah, I shouldn't say high. I said elevated, right? So the pH of urine it ranges from 5.5 to 7.4 in their assay.
Yes,
Okay.
and in most urines rarely go above 7.2, I think.
Okay. So how do you prove that aryl alcohols do it? What did they do?
They literally went back and they added them in.
Yeah.
And there's a table that they have, the aryl sulfate associations with restrictive urines, and they have mass to charge ratios and fold higher in the restrictive urines. And if you look for significance, it's the C11H9O6S that had a p-value of 0.0004. So as they begin to take you through that sulfol metabolite in your urinary pH, and they, figure 10 of their story is the one I would commend the readers to carefully studying, because it's a multiple logistic regression analysis of siderocaline activity shows independent association between the siderocaline activity, which we make to control UTIs, and pH or the sulfol metabolite composition. And then they have this beautiful bubble plot of all 87 of the urine specimens tested in the study as a function of pH and aryl sulfol metabolite. And they again make this new DA score where the radius of the bubble equals the siderocaline activity of the specimen. And it's just an absolute fascinating way of looking at this whole notion of urinary pH and what restrictive and unrestrictive urine is. And they introduce you to the highly restrictive characteristic of the siderocaline activity based on your urinary pH and the presence of this protein. And I think the key to these 5 series of figures is they had the keystone is the ROC curve, or the receiver operating characteristic curve, which is a plot of the true positive rate against the false positive rate. And again, this is why it's a interdisciplinary project, because they really made some sense out of this very important infectious process, but they took the reader through something very complex in a very logical, stepwise manner of taking you through the approach to put the thing together.
So the last 2 panels of that figure are really the key, where they're growing the E. coli in permissive urines and showing if you add aryl alcohols and bicarbonate, you inhibit their growth. They're gone.
It's so elegant.
So the permissive urines have the problem that their pH isn't quite high enough, and they're lacking in aryl alcohols, and you could see that by that interesting part C there, that graph that you were mentioning.
Right.
So they would find in the permissive area towards the bottom where there are not enough of these metabolites.
Correct.
Pretty cool.
And so
what's the model? What's the model then, Michael? What's all this doing?
So the model, they take you through the story. Your bladder lumen is infected with E. coli because it's been inoculated because of our bad plumbing. And the net consequence is the urothelium then produces this siderocaline.
Right.
E. coli is constitutively producing enterobactin.
Right.
And siderocaline and enterobactin are having this war.
Right.
And if the pH is right and you have these aryl metabolites, you can actually make your urine restrictive or permissive in the presence of E. coli and enterobactin, because enterobactin is good enough to steal iron away or prevent the siderocaline from doing what it's supposed to do. And as they take you through their proposed model, in panel A, they say human urine contains a complex and variable mixture of metabolites, including these aryl metabolites such as catecholates. Iron is always limiting because we're it up, and it exists as a labile pool of low affinity complexes with numerous ligands. And so some of these metabolites, because catecholates can also bind iron, and the aryl metabolites are sort of siderophores that are binding iron as well, and at a lower affinity.
And that's all facilitated at a higher pH.
Correct.
So the aryl metabolites, as you say, are acting sort of like chelators of iron together with the siderocalins, and that can steal the iron away from the enterobactin.
Mm-hmm.
But if you have just the siderocalin alone with the lower pH and without the aryl metabolites, it's not as good as taking the iron away from the enterobactin.
Right. And that's, I think, what they did is they put it all together with urine samples as opposed to a bladder model that the other workers in the field had used before. Yeah. And so the elevated pH facilitates the host-derived ferric aryl complexes in the siderocalin calyx, starving the uropathogenic E. coli of iron.
So these aryl alcohols, Michael, are normal metabolic products in the bladder,
correct,
right?
And yes, so from our gut, and they're getting dumped into our kidneys,
right?
And they're coming out.
So some people have good levels, and some people don't. And what is that dependent on? Do we know what controls the level of aryl alcohols? It could be diet, right? It could be genetic. It could be environmental.
Yeah, and in fact, this working model suggests, and this is the quote, suggests important areas for future investigation and opportunities for new virulence tagging UTI therapeutic approaches distinct from broad spectrum antibiotics.
So in the simplest, crudest sense, you could just drink aryl alcohols and hope they got to your bladder, but of course that's probably not going to work, and it would probably not be good for you.
Your liver will probably process it to nothingness.
But if you found out that, for example, carrots, just to pull something out of the air, raise the aryl alcohol levels in your bladder, that would be something you could do, right? Or maybe asparagus, Michael. I know that some asparagus metabolites get into your bladder for sure.
and maybe even cranberry juice, because when I had my kidney stones a while back, the urologist said drink lots of cranberry juice, and being the curious individual that I am, I asked him why, and he says I don't know, but it works.
Yeah, so they say urinary catechol are associated with tea, coffee, wine, and cranberries.
Yeah, I think I'd rather drink the wine.
Yeah, so you'd have to do that in the evening. You don't want to be drinking it for breakfast, though.
No.
Because then you're not productive, right?
Then you're not productive. You wouldn't get through this 11-figure paper.
Oh, so it could be something as simple as diet or something more complicated. So how do you, let's say it's not just diet that could raise your urinary pH and aryl alcohols. What other approaches do you think? You actually injecting something into someone, but it may not get to the bladder, right? That's tough.
I think the traditional way is the solution to pollution is dilution. So they always tell you just drink more water if you're concerned about urinary tract. And the other way they do it is they aggressively modulate the flora, and some people have encouraged the consumption of yogurt. So the lactobacilli change the vaginal vestibule's flora by lowering the pH. Some have prescribed flushing to change the pH, but it's a lot of these urinary tract infections are a consequence of antibiotic therapy, where women go on antibiotic therapy and they get a yeast infection, which is distinct from a cystitis, which is an infection of the bladder.
So that's why this is a nice lead that you don't need. It's an alternative to more antibiotics, right?
Correct.
Using naturally produced compounds, figuring out how to induce them, and going from there. And
or maybe just going and doing the experiment. The way you could do it is tell people to take, in women who are susceptible, that is, they've had more than their fair share of urinary tract infections, you could do a simple experiment of looking at these aryl alcohols. Can you modulate your diet easily by cranberry juice or wines or teas to see whether or not you could effectively modify the behavior of the host such that it would change. And, it would be an easy experiment to do. You'd say, okay, did you eat the following things? Please give me your urine sample. And then with their ratio, they will be able to determine permissive versus non-permissive. And were you able to change your urine from a permissive urine to a restrictive urine?
Yeah, you could set up a clinical trial where you have people drink cranberry juice and collect urine and check out the permissivity of the urine. It's not invasive at all, right? So it's not a hard study to get approved.
No, and I think that's, again, why I spent so much time going through that second figure, is because it's that beautiful set of mutant versus wild type that enabled them to really, I think, give us this interesting result.
I wonder if the Yanomami get urinary tract infections.
They probably do. Their plumbing's really no different than ours because they're classified as humans.
But maybe they have elevated pH and aryl alcohols for some dietary or genetic reason. Who knows, right?
Yeah.
Anyway, I corresponded with the first author on this paper, Robin Shields Cutler, who's a guy.
Okay, he's Brit?
No, he's an American.
Oh.
He's raised in Stillwater, Minnesota. Not too far from the Twin Cities, right in the border with Wisconsin. He went to Grinnell College in Iowa, got a BA in biological chemistry. So he's published in the Journal of Biological Chemistry, BA with honors. I asked him how he got interested in science, and he said he always was interested about how things work, and he liked problems that he could use his hands to fix them, like repairing bikes, cars, and stuff around the house. So he didn't have any real exposure to science, but he just liked fixing things. And he got to high school, and he had some great science teachers who taught him how you could have science as a career, and they encouraged him. And the more he thought about this, the more he got interested in host-pathogen interactions. And he gravitated towards biochemistry because of his interest in, mechanical things, fixing things. So he went to college, and as I said, he majored in biological chemistry. And he ended up in the lab of Jeff Henderson, who's the PI on this paper. And he had just started his lab when Robin started graduate school. And he liked his enthusiasm. He liked his chemical perspective on pathogenesis, which is, if you listen to TWiM, we see that in a lot of papers using chemical approaches. And he said that using analytical mass spectrometry combined with basic micro is a great way to understand small molecule chemistry. And that resonated with him. And he thought the siderophore issues and siderocalins and UTIs were very interesting. So he wanted to work on that. And then turned out he thought Jeff would be a great mentor. I guess that's the sort of it's just sort of stuff you have to pick up by talking to people and getting to know them a bit, that you say, yeah, this person could be a good mentor for me. I asked him if there were any surprises or challenges during this work. So this was his thesis work. The first author on this paper. It's his PhD thesis. In fact, he defended, the day before yesterday, June 30th.
Well, that's good.
Yep.
Hopefully it was a success.
Yeah, it is. And he said he was amazed at the magnitude of differences between individuals in the urine activity, which we said that's something that I was struck by as well. It was surprising that urinary composition had such immense control over the efficacy of siderocalin. A lot of really great work had previously described the protein's antimicrobial activity, but this influence of the urinary context hadn't been studied. Of course, using individual human specimens presents a number of challenges due to variability, but using mass spec and other biochemical techniques, we use the differences as a variable to discover more about this antibacterial response. So that's an interesting way to put it, right? You have person-to-person variability. You use it to tease out what's going on, not get frustrated. We were also surprised to see the typical siderocalin-enterobactin relationship inverted in urine. In other models and host contexts, it's understood that siderocalin effectively neutralizes enterobactin. The urinary context of some samples, as we showed, essentially reverses this, and enterobactin really emerges as an important bacterial factor to overcome siderocalin. Again, the magnitude of this effect varies, but the correlations with pH and a class of dietary source metabolites give us some hints about where this research could go. So these metabolites are dietary sourced, as we've mentioned. So that's, as you said, might be one way to go and help people. And I asked him what was in his future, so he's just offended. And he's going back to Grinnell. How do you say it, Michael? Grinnell. He's going back to Grinnell, where he was an undergrad, for a 1-year position as an assistant professor of biology, and then he's going to do a postdoc after that. So that's an unusual course, right? I guess it's kind of cool. You work for a year. I suppose he's going to do some teaching, and then do a postdoc.
Well, Grinnell is. I have a collaborative research project with individual at Grinnell. And we've been doing some neat things, and she's got a group of undergraduates that work with her. And she got her PhD did her postdoc, I can never keep them straight, from Jim Teege, and she's now on faculty at Grinnell. And her name is Shannon Hinsa-Leisure. And so we've been doing collaboration there. So I periodically get out to Grinnell. So I may have an opportunity to meet him.
Look him up. Anyway, great paper, Robin and Jeff and all the collaborators. And thanks, Michael, for doing that. We got a couple of emails to read here. First is from Jim. For your interest in listening to physics, there is a podcast called the Titanium Physicists Podcast. They use a conversational format similar to what you use. They have a panel of experts and a guest who is not a physicist, but who is accomplished in some other area. By the way, I recommended the Twix group to him when he was looking for biology podcasts. Love the 4 podcasts you people do. So I'd mentioned somewhere, I guess on TWiM, that I'd love to hear a physics podcast. So I have subscribed to the Titanium Physicists Podcast. I'll let you know how it is. Thank you, Jim. Lisa writes, hello, TWiM overlords. I have been listening to this podcast since it first came out in 2011. Now I'm an incoming senior at UC Berkeley, majoring in microbial biology. It was your podcast that helped me decide what to study. I would also like to say hi to the graduate student that captured fruit flies with wine. Perhaps we have already met. I think that student was from Berkeley as well. It's summer for me, and I've been re-listening to the podcast. Everything is glorious, but what caught my attention is TWiM 48. It's all about direction with Joe Handelsman. This was released in 2013, and it talked about the deficiency in science-educated workers at the bachelor's level. I was hoping in working in a startup or an industry for a year or two before I went to grad school for a PhD, but should I bother at all? I don't plan on going to academia, and I don't know what to do about further education. My grades are not the best, but I'm a student researcher in a lab up in the hill, and I enjoy research. I have a lot of autonomy in what I can do, and I'm afraid I'll never regain that sense of freedom without a PhD. At the same time, I don't want to get lost in constant paperwork. Berkeley has a lot of academics, so I don't know what it's like in industry. I do enjoy when you guys bring it up in the podcast. This podcast was the fodder to keep me going through bad semesters. At first, they all seemed fascinating and quite confusing. Now I understand more, but more importantly, I'm questioning the paper and thinking about what the data means. I still have a long way to go, and in a way, I feel like I have learned nothing because there's always so much. I would like to thank all the hosts of TWiM for being part of my high school and college experience. I still have a year left in college, but I should have thanked you 3 years ago when I first got accepted. Well, I would say, Lisa, so you're working in a lab. You know what research is like. I don't think it's a bad idea to work for a year or two. I did it. I got out of college, and I worked for a year in a company doing microbiology because I wasn't sure what I wanted to do, and I loved it. And I knew from that alone that it was what I wanted to do. Now, I don't know if you know already from your undergraduate experience if you know that research is for you or not. And if you don't yet have that sense, work for a year or two. What do you think, Michael?
I think it's great advice. I too worked a year between undergraduate and graduate school, and it's really a great opportunity to figure out what you're really passionate about. It really is a function of getting the right boss, and most people who are in positions of leadership or supervisory roles generally will give you opportunities to go out on your own if that's the mission of their group. If you get into a QC group where you're doing quality control, you have to do it by the numbers. But if you're in a lab that's pushing the frontiers, generally, if you're curious and you have good experimental reasoning or good thought processes, they'll let you run with it. And that's what really research is all about, is, thinking about what's next and how best to answer the question.
It's funny, Michael. My 1-year job between college and grad school was in a QC lab.
Uh-huh.
And I liked having protocols, because I could learn them. I could do them really reproducibly. I loved that idea that I could do something over and over and it would work. On the other hand, I said, I don't know anything and I've not learned enough here. I need to go back to school. So the QC was perfect in that it showed me how science could be reproducible and rigorous, yet it showed me that there's a lot more that I needed to go and find out. So for me, it worked.
Yeah, and there are a lot of important jobs out there that, really add value to society. This past week, we had a 2-foot water main break in downtown Charleston, and the water pressure dropped in the peninsula. And so we had a boil advisory everywhere on campus, in the city, and it required the water department to issue an all clear. And so how soon can you know that your water is safe to drink, that you don't have to boil it to do something simple like make coffee?
Yep.
And, that was a bench level tech who had to, collect samples of water from throughout the city to see whether or not our water was safe to drink after that pressure drop.
All right. So basically, Lisa, I think it's worth working if you're not sure. If you're sort of, because you really need to get into science as a PhD, you really need to have total passion. If you're not sure yet, just work for a couple of years. I think it's fine. It is absolutely worth it. Next letter is from Kathy Spindler, who's a co-host on TWiM. Too bad Elio's not here. This has to do with the California drought. In the late 1970s, when there were a couple of years of droughts in California, the complete saying was, if it's yellow, let it mellow. If it's brown, flush it down. That's how many, it's right. It's urine in the toilet, right?
Oh, yeah. And it's appropriate for this episode this week.
Perfect. And a very memorable billboard I saw in the Central Valley showed someone in the shower with the caption, "Sing shorter songs." I enjoyed crossing over to TWiM 104 to hear the measles story covered. So that was the story that you snippeted, because it came up on TWiM and they said, should we do this? I said, well, Michael grabbed it already, so go listen to TWiM.
Well, and I turned it into a qualifying exam question for our graduate students.
Nice.
Yeah, it was a great qualifying exam question.
I often do that. I have turned news items into exam questions. Yep. Steve writes, hi, Vincent and Al. I was just listening to TWiM 41 interview with the health map man. Oh, yeah, I remember that.
Yeah.
Do you remember, Michael? That was, I don't know what city it was in, but that fellow from Harvard who had a role in health map called it up to have a look. There were a couple of red dots not far from me, one a mystery illness making an EasyJet plane turn around, and the other this shocking news about the endangered saiga antelope. It does not seem that long since I was watching wildlife programs that featured migrations of seemingly limitless numbers of these animals. Now we hear that they are being wiped out with extraordinary speed, but by what is thought to be a bacterial infection. It seems to be happening almost impossibly fast. Perhaps the team would like to look into it for your podcast. So he sent a link to a Red Orbit news item. Mystery disease claims half of endangered antelope populations. Carl Zimmer also wrote about it in the Times. Saiga antelope mystery disease die-off. Apparently, it stopped, and they don't really know the causative agent, so not much we can go on there. Hopefully,
and they didn't do any necropsies of the antelopes to figure out if it was a weird infectious agent.
Well, they did find Pasteurella and Clostridium. So they were infected with those two. I don't know how many, but they suspect that something else contributed because these two bacteria are common in healthy animals.
Yeah.
And it's only a problem when the animals are sickened. So they did, but they didn't find anything else. So the answer is we don't know. what's doing it. And it possibly will need to have another outbreak. 250,000 animals died in 2 weeks.
That's a lot.
It's in Kazakhstan. So, I need a paper to go on, Steve. And there's no paper. Not yet, anyway. Steve is from England. Weather still rather gray and windy, but I don't get out as I'm housebound with ME and can only stay upright for a few minutes at a time. Very sorry about that, Steve. But I'm glad you're listening to TWiM. Wink writes, Michael, loved your SNP, SNP. Now, I presume he's kind of making a joke about snippet and snip.
Yes.
In addition to washing produce, only reverse your stomach's hydrochloric acid when necessary. Also, the STEC dose is lower than salmonella. Remember the washing produce? Now, were SNPs involved in that to identify the strains or something? I don't remember.
I have to go back and listen. They're all running together now. But we're up to 107.
What episode was that? With the
here
recent. We go. An assessment that was 105. An assessment of the microbiology safety of fresh whole leaf herbs from the UK.
Oh yeah, that's the UK one. Yeah.
So I don't know. I don't recall that.
It wasn't SNPs. It wasn't SNPs involved.
Maybe
it was just pH.
Maybe he's just saying snip as a cute way to say snippet, but snip has other meaning, of course, looking for single nucleotide polymorphisms. So we are slow on the uptake, wink. All right. What is the business about hydrochloric acid in the stomach, Michael? Is that a joke? In addition to washing produce, only reverse your stomach's HCl when necessary.
Well, we know that because of all the press coming out about the proton pump inhibitors, and HCl is very important for cardiac health, it seems now.
Okay. Got it. All right, last one's from Stuart. Greetings from sunny Seattle, where it's 21 degrees C with clear skies.
Ooh, that sounds nice.
Because you asked, here are my 2 cents on the whole hand-washing situation that has been discussed over the past few episodes. My solution would be to change building codes to require shared sinks for men and women that are located outside the actual bathroom. This would ensure compliance because it would allow for policing without violating any privacy issues. It could be especially effective in a restaurant setting where a manager could simply discipline employees they watch leave the restroom without making the required stop at the hand-washing station located outside. Additionally, some peer pressure and social stigma would be enough to guilt people into not skipping the hand-washing station. Behavior might change if poor hygiene is on display for everyone to see. Thanks for the great podcast. I'm a fanatic TWiM fan and listen to the podcast on my 1-hour commute to my job in a manufacturing lab at a biotech company. Long-time listener, first-time emailer.
Well, that was nice.
I don't know about moving sinks outside. That would only work if you have the 2 bathrooms close and you could have a common outer room, right? And I'm not sure that people would like that. I think it would be costly. And I'm not sure having everyone out there. I mean, look, I go to a men's room. There are lots of guys there. I don't need to have women there to make me guilty into washing my hands. Of course, I wash them anyway. But I would think just having people around. But a lot of people are not susceptible to that. Now, of course, it allows policing. You're right. But, boy, then it gets complicated. I just don't know if that is sensible or makes sense monetarily.
Yeah, it's. I think it's it's up to you. It's up to you as the tribe of Venezuela has shown us. You'll just get bigger microbial diversity if you fail to wash your hands. But unfortunately, we have antibiotic resistant creatures in our facilities as opposed to what they have in theirs.
Yeah. This all began quite a few episodes ago with someone who questioned having employees required to wash their hands. And
that was the senator from North Carolina.
And so I'm glad that we have continued this discussion. I do think the signs are fine. If you are a libertarian, I understand where you might not want to tell people what to do, but I think it makes sense. Even if it's not 100% effective, it's still going to make sense. And if we can get some, it's not an easy answer. I don't know what the solution is, but taking away the signs, I don't think is the answer to this problem. Should we leave the signs in, Michael?
Leave the signs in.
Of course, it's not up to us, so Who cares?
We're not in charge this week.
All right, that's it for TWiM. This is TWiM, This Week in Microbiology. TWiM is Fridays. Number 107. You can find them all at iTunes. You can find them at microbeworld.org/TWiM. You can find them on your podcatcher, which is on your Android or iOS device. We're just everywhere, and they're all free. The whole archive is there for you to listen. Some of the back ones are cool. Check them out. And we like to get your questions and comments. You should send them to twim@twiv.tv. You're my co-host. My co-host today has been Michael Schmidt from the Medical University of South Carolina in Charleston, South Carolina. Thank you, Michael.
Thank you, Vincent. Have a good holiday weekend.
You too. Happy July 4th. Be careful. Safe travels.
I will try.
I'm Vincent Racaniello. You can find me and everything I do, podcasts, blogs, lectures, all at virology.ws. And I do want to thank the American Society for Microbiology for supporting TWiM, in particular Chris Condain and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.